Hello, my dear friends. Welcome to Live Prep. Myself, Dr. Gajapati Verma. As I informed you earlier, under the name of Bootcamp, we are providing both revision lectures and also we are conducting test series. Revision lectures are available both on Live Prep app and also on Live Prep India YouTube channel. But test series can be given only through the app. Clear? And now let us quickly go through the revision of the chapter Locomotion and Movement. The chapter, please remember once again I am telling you, we are confining ourselves only to NCERT content. Clear? We are not going out of NCERT. So now let us quickly go through the content of this particular chapter. So first they are talking about some movements. So here first they are trying to tell us what is the difference between movement and locomotion. So in this way they are giving some examples. Let us quickly talk about this. The streaming of protoplasm or protoplasmic streaming, it is also called as cyclosis, that we are observing in the eukaryotic cells. So this is also an example for some movements. Yes or no? There is also example for movement. This is what they are saying. But here in NCERT they are talking within the amoeba. Please remember, this is not just happening in amoeba, it is occurring in all eukaryotic cells. Okay. Next one, the movement of cilia, flagella and tentacles by different organisms. So different organisms are having these structures. So they are able to move. So when they are moving means they will come under the example of movements. And please remember, wherever the cilia, flagella and tentacles are present, don't think that always the cells having these particular structures are able to move. Please remember, the cell may or may not be able to move, but these structures are able to move. Please remember this. Okay. So what I am trying to tell you is, let us consider a particular cell that is having cilia. Let us take an example of paramecium. Paramecium is having cilia, and with the help of cilia, so cilia are moving anyhow, and because of this movement of cilia, paramecium is able to move. So in this case, the cilia containing cell is able to move. This is one example. Other side example, let us talk about our trachea. So in our trachea also ciliated cells are present. Cilia are able to move and they are pushing the dust and mucus towards the pharynx. So here cilia is able to move, but the cilia containing cell is not moving, that is present at that location only. So in this way, in general when we are talking, so the ciliated cells are able to move is a general statement, but not always. So depending on the location and all the things, you need to understand these particular points. Anyhow, regarding these particular things, already in the cell chapter itself we have covered. In the regular chapter we have covered, and when we are going through the revision also, at the time also I gave you some examples. Okay, clear. Next one. So human beings, if you are talking about the human being, so we are able to move our limbs, jaws, eyelids, tongue and all these things. So we are able to move all these things. So the movement of all these things are examples for some movements, body movements. Okay. So this is what they are saying. Next one, some of the movements that may result in the change in the place or location. Suppose I am here. So now my position is not changed. I am here only, but I am able to move my hands and all the things. Yes or no? But if my position is changed, there are some movements. This is also a movement, the movement of the legs that is able to cause change in the location. So in this way what happens, some movements are causing some voluntary movements. Please remember. So why this word called voluntary is very important. Now let us see. Let us consider that there is a bacterium. There is a bacterium and the bacterium is not having flagella. Please remember, this content I am linking with cell chapter. Let us consider a bacterium and the bacterium is not having any flagella. Please remember, in the case of bacteria, when you can say that it is motile, if it is having flagella, then it will be motile. Yes or no? If flagella are not present, bacterium is non-motile. Yes or no? So now I am taking that example, bacterium, but it is not having flagella. But this bacterium is present within water. Suppose the flowing water is there, river water is there, in that also bacterium is there. Now because of this movement of river water, the bacterium is able to move. But please remember, in that case you are saying that bacterium is non-motile because it is not able to move on its own. Since it is present in a flowing water, it is also going along with it. So that you should not consider. So that's why they are very clearly saying that such type of voluntary movements, if I want to move on my own, then I should be able to move. So these are voluntary movements. Only such type of some voluntary movements can result in change in the location or change in the place of an organism. So that type of movements are only called as locomotion. Please understand this. So in this way, they are trying to give us some examples here. Suppose there is an organism or animal or something is there. So that is walking. Walking means what? Its position is changed. Running, climbing, flying, swimming. In all these examples, what happens? The organism location is getting changed. So that's why all these will come under examples for locomotions. Understood or not? So in this way they are saying, and then finally they are giving some examples here. So anyhow, you need to understand one conclusion that all locomotions are movements. Yes, all these locomotions are because of movements only. But all the movements cannot cause locomotion, and they are not resulting in locomotion. So this is what one important conclusion that they have given in NCERT. So try to understand these with examples. Clear? And here they have given some other examples. What are these examples? If we are talking about the paramecium, in the case of paramecium, so cilia are present. Very good. But here what they are saying, the same cilia, now here. So here it is the cytopharynx, and here it is helping in movement of the food through this particular cytopharynx. The same cilia, now here, now it is also able to cause locomotion of this particular organism. So in this way they are giving some examples here. The cilia that are present in paramecium, they are helping in movement of food through cytopharynx. Not only this, it is also helping in locomotion. This is what they are saying. So the cilia are playing different roles. They are causing some just internal movements, and they are also helping in locomotion. In the same way they are talking about the hydra. In the case of hydra, it is having the tentacles. Now these tentacles are used for locomotion anyhow. Not only this, these tentacles are also used to capture the prey. So in this way it is having different roles. That's what they are saying. And in the case of human beings, if you're talking about the limbs, mainly with respect to this lower limbs, legs. So now these, so we can, with the help of these limbs, we can change our body posture. That is one thing. Second thing, we can also able to move from one location to other location. So they are also involving in locomotion. So in this way, they are talking about some examples. So in this way, there are some structures that are having they are not only involved in locomotion, but they are also involved in some other functions. This is what they are saying. Clear? So in this way, whenever we are talking about these locomotions and movements, we cannot talk them separately. So both of them are interlinked with each other. That's why we are talking about all these things. Then they are talking about what are the different types of movements that we are observing in cells of humans. So now observe carefully. This is with respect to the cells of movement. Now here they have given in NCERT, they are talking about three types of movements. One is amoeboidal movement, next one is ciliary movement, and the next one is muscular movement. So amoeboidal, please understand this. Amoeba itself is showing this type of movement. That's why it is called as amoeboidal movement. Then how amoeba will move here? Amoeba will move with the help of pseudopodia formation. And regarding the pseudopodia formation, much information was not given here. So that's why I'm not going into these details. During the pseudopodia formation, what happens? Cytoskeletal structures will play very important role. Please understand this. So the cytoskeletal structures, now they are undergoing some rearrangement inside the cell. So with the help of this, some pseudopodia will be formed here, and hence this amoeba is able to move. In the same way, the way how amoeba is able to move, in the same way, there are some cells in our body like macrophages, leukocytes, and these are also able to move inside our body. This is what the comparison they are saying. So in this way, these cells are able to move like amoeba. So that's why this type of movement is called as amoeboidal movement. Please understand this. Next one, ciliary movement. There are some cells, they are having the cilia. But please remember, these cells in these movements, please understand, these cells are not moving. Please understand this. I will tell you with example also. In the case of trachea, so it is a part of respiratory tract, yes or no? So in the case of trachea, what we are saying, there are some cells that are having cilia here. They are having cilia here. Now what happens when we are taking this breath? So breathing process, already the respiration chapter already we have studied, right? Breathing and exchange of gas. When we are taking the air in, now what happens? Along with the air, some dust is also coming here, and now this dust should be filtered here, and now this dust, and sometimes some mucus is also produced. Now this dust and mucus, now they should be thrown towards the pharynx. Yes or no? They should come towards the pharynx so that we can spit it outside. So in this way, the cilia will help in removal of dust and also the movement of mucus here. But the point here is cilia are able to move here. But the cells that are having this cilia, these cells are present there itself. The cells are not moving in this case specifically. Okay, please remember here. And another example they are talking is with respect to oviduct. So oviduct is actually the part of female reproductive system. Female reproductive system. So in the female reproductive system, so we are talking about the oviducts here. So oviducts we are talking, and these oviducts are also called as the fallopian tubes. These are also called as fallopian tubes. So this is the ovary. This is the ovary. Okay. So this is the ovary. Now from this ovary, the ovum is released into this coelom, and from this coelom, now it will be captured by this oviduct. Anyhow, I am not going into the details of this oviduct, so please observe carefully. And now this ovum, ultimately it is entering into this oviduct or fallopian tube. And now this ovum should move towards the uterus. This is what the uterus, right? The ovum should move towards the uterus. And now you need to read the question and options very carefully. How this ovum is able to move towards this uterus? Now we are saying that generally, in the general sentence, what we will say? So it is the cilia that are helping in the movement of ovum towards the uterus. Correct? Correct. But technically you need to understand the question here. Now where these cilia are present? The question is very clear, so please understand. Many students are making mistake here. They know that cilia are helping in the movement of ovum towards the uterus. That is correct. But where these cilia are present? Does this ovum has cilia? Does this ovum has cilia? No. Ovum does not contain cilia. So please understand the points with clarity. Then who is helping here? Now this is the fallopian tube. And now on this fallopian tube there are some cells. And actually these cells have the cilia. The cells of fallopian tube, actually they are having this cilia. Please understand this. The cells of fallopian tube have the cilia. And now this cilia will help in movement of ovum towards the uterus. So please understand this. Ovum does not contain cilia. Ovum does not contain flagellum. Whereas the cilia of the cells present in fallopian tube, they are helping in the movement of ovum towards the uterus. Don't make mistake. I am observing that many students are thinking that ovum is having cilia. That's why it is able to move. No, ovum is not having any structures like cilia and flagella. Please understand this. So don't make mistakes. So in this way, there are some cilia containing cells. They are helping in some other process. That's it. So these examples are like this. And that too, this example of oviduct is applicable only for female organism. With respect to human, if you are talking, it is applicable to female only because it is not present in the male. This is also another important point that you need to remember. Next one is regarding the muscular movement. There are different types of muscles are there. They are helping in the movement of various organs. Now here in general we are talking here limbs, jaws, tongue. These are the examples given in NCERT. Likewise many organs are there. Now at these locations muscles are present, and these muscles are able to move so that they are able to cause the movement of various other organs that are associated with these. So this is what some general example that they have given in the NCERT. Now along with this, in the separate paragraph, they are talking about flagellar movement. There are some cells that are having the flagella, and these flagella are also helping in the movement. With respect to human, if you are talking, it is the spermatozoa or the sperm cells that are having the flagella. So please remember, human male is having these flagellated cells. Sperm cells are the flagellated cells. So human male has flagellated cells, but flagellated cells are absent in human female. Very, very important for examination. So please remember this. This is with respect to human. Now overall, if you are observing, there are some other examples like coinocytes in the sponges. So in the sponges, when we studied about the fungi, so there we studied about this. So coinocytes. So actually they are present here at this cavity, and actually they are helping in the movement of this water. So that's why they are helping in the movement of the water here. So that's why they are helping in the water current formation here, the movement of water here. So in this way also, these flagellated cells called coinocytes are helping in the movement of water in the case of sponges specifically. Next one, in the case of euglena, here you can see the presence of flagella. So now this flagellum is helping in the movement of euglena. So in this way, even the flagellum is also helping in the movement of some cells. In this case, cells are not moving, but the flagellum is causing the movement of water that is present surrounding this. So in this way, please understand cilia and flagella. Please understand very carefully. Don't think that always the cells having these particular cilia and flagella are able to move. That is wrong concept. Sometimes they may be able to move like this, and sometimes they may not be able to move. So please understand these points. Okay. So these are some examples that they have given in the NCERT. Then they are saying that, so finally some general conclusion they are saying. So what are the locomotion? Locomotion is the change in the location of the organism, right? So this locomotion requires coordination of various things. What are required here? Coordinated activity of muscles are involved here. Skeletal system, so bones, cartilages, they are also involved here, and the neural system, because the neural system, from the nervous system we are getting the information or the signal for the movement, and this signal is ultimately going to the muscles. So muscles are involving in the contraction, and these muscles are attached to skeleton. So in this way, ultimately the skeleton is also able to move. So ultimately it is resulting in change in the location. So for the location, so all they should be highly coordinated. This is what the sentence and its meaning. And now we will talk about the muscle. What is in general, what are the properties and what are the different types. Likewise they have given. Now let us quickly go through this. So muscle, so it is a mesodermal in origin. Next one, 40 to 50% of the body weight is constituted by muscle itself, and it has some special properties like excitability. What is the meaning of this excitability? Please understand this. Actually, this excitability concept you are studying for the neuron under the neural control. Anyhow, in the next next revision part we are studying that one only. So with respect to neuron, what we are studying, some electrical potential is changed across the neuronal membrane, right? And that itself is called as excitability. If there is an impulse, according to it, it will undergo excitation. What is the meaning of this excitation? Potential change will happen across the membrane, that is the plasma membrane. So same way, whatever we are studying for the neuron, in the same way for the muscle cells also, these potential changes can occur. That's why we are saying that muscle cells also can undergo excitation. So excitability is the property of both muscle cells and neurons. Don't make mistakes. So please understand and interlink the concepts. Next one, contractility. So muscle cells can undergo contraction, but not the neurons. So in this way, the length, sarcomere length, anyhow we will study in the next coming sections. So sarcomere length will decrease upon contraction, and upon relaxation it will come back to normal. Okay, this is what they are saying. Extensibility and elasticity. So that's why the entire muscle can undergo. So it can undergo shortening and it can again go back to the relaxed state. So in this way, when compared with the shortening, so relaxed state is considered as elastic or extended state. So in this way, they are saying that the muscle cells can undergo contraction and they can also undergo extension. So these are the some points that they have mentioned. So please observe carefully. Next one, we are classifying muscles based on different properties, based on the location, based on the appearance, and based on the nature of regulation. We can classify muscles into different types. First we will talk about the first one, based on the location. If we are observing the location, based on the location, muscles are three types: skeletal muscles, visceral muscles, and cardiac muscles. So all the three at a time I will try to tell you, so observe carefully with comparison. Skeletal muscle. Why it is called as skeletal muscle? Because this muscle is attached to skeleton. Skeleton means skeletal. Under the skeletal system, what they have mentioned in NCERT. Under the skeletal system, we are talking about the bones and cartilages, right? Because they are very important for the basic support of the organism. That's why it is called skeleton. So these muscles are associated with the skeleton of body. So that's why these muscles are called skeletal muscles. Next one, visceral muscles. Generally, these are present in the visceral organs like digestive system and reproductive system. Likewise, at many systems, these muscles are present. Okay, that's why it is called as visceral muscle. And next one is cardiac muscle. So it is present within the heart. So that's why it is called as cardiac muscle. So in this way, based on the location, we are classifying muscles into three types. Please understand. So sometimes they may say that muscles are three types, like these names. They will give these names. But they may ask in the question that what is the basis for that? What is the basis for that? Location. So that's why these points also you need to remember for examination. Clear? Now let us come to the next one, appearance. Based on the appearance, see here, some muscles are having some dark and light bands. If dark and light bands are present, it is called a striated appearance or striations. So some muscles are having striations, some muscles do not have the striations. If they do not have striations, they will appear smooth, continuous. So that's why here we are using some names called striated, non-striated. Now, based on the appearance, who are appearing as striated? Skeletal muscles are having alternate light and dark bands, so that's why they are striated muscles. Not only this, even cardiac muscles are also appearing as striated. Clear? But visceral muscles, they are not striated, they are non-striated. So non-striated is also called as a smooth. So that's why visceral muscles are also called as smooth muscles. Clear? So that's why please understand, visceral muscles are also called smooth muscles. In the examination, if they are talking about smooth muscles, means they are nothing but visceral muscles. But here there is a great confusion. What is that confusion? Both skeletal and cardiac muscles are striated. But sometimes, please understand, you need to, that's why you need to understand this question and in what context they are asking. So please observe carefully. But in general striated muscles means skeletal muscles. In general, striated muscles means skeletal muscles. Please understand this. Even though cardiac muscles are also striated, but generally the term striated muscles we are using with respect to skeletal muscles, because generally for this we are directly saying as cardiac muscles. Clear? So please understand this. Next one, nature of regulation. Whether the activity of these muscles is regulated under the will of an individual or not. If it is under the control of will of an individual, that is called as voluntary. Suppose I want to contract this. Yes, I can contract this. I want to relax this. So I can relax this. In this way, if the contractions and relaxations are under the control of will of an organism, then it is called voluntary. If it is not under the control of will of an organism, then it will come under involuntary. So in this way, only the skeletal muscles are under the control of this will of an individual. That's why these are called voluntary muscles. So contraction relaxations are under the will of organism. But these two, visceral and cardiac muscles, these are not under the will of organism. So that's why these are considered as involuntary control. So these are involuntary. Is it clear? So please understand this. This is a very, very important table. Questions can be asked in any way, and directly assertion region questions can be asked, or match the following can be asked. So direct statements also can be given. So please observe this. This is a very important table. Now, along with this, we also need to understand about some other small properties, not clearly mentioned about these in NCERT, but please understand this. This is the diagram from the old NCERT textbook, but this diagram was given in animal tissues, but it was deleted in the new NCERT, but simply diagram based. So observe carefully. So this is for skeletal, and this is for the visceral or smooth muscles, and this is for the cardiac muscles. Simply based on the diagram, you please understand this. So skeletal muscles, they are cylindrical structures. Even the cardiac muscles are also cylindrical structures. Understood? The comparison wise, you just go it. Next one. In the case of cardiac muscles, branchings are there. It is branched. Cardiac muscles, now here the cells are branched here, but here these are not branched. These are not branched. So these are some points that we need to understand. Next one, already we studied that there are some striations are present here. So striations are present here also, but we are not observing striations here. Understood or not? These are some important points. Next one, cylindrical, but here if you are observing, so here at the ends it is tapered. Yes or no? Ends are pointed. So this type of structure is called a spindle shape. So at the center it is a little bit wider, and at the ends it is having pointed ends. This type of structure is called a spindle shaped or tapered ends are present here. Clear? Next one, striations are not present. That's why it is called smooth. Is it clear? Next important point. So anyhow, these two are involuntary, this is voluntary, that's okay. But the another important point is, this is completely one cell, and one cell is having one nucleus, and here also one cell is having one nucleus. But in the case of skeletal muscle cells, please observe carefully. For the skeletal muscle cells, this is considered as a syncytium. What is this syncytium? In the regular lecture under the cell chapter, very clearly I have explained about this one. What is this syncytium? Syncytium. Okay. Syncytium means syn is the fusion. Simply understand this one. Okay. Syn is the fusion. So fusion of the cells. So initially suppose there is a single cell with single nucleus. There is another cell with single nucleus. Okay. But when these two cells are fused, now what happens? The plasma membrane will fuse together. The plasma membrane will fuse together so that it will become a continuous one. Understood or not? But here the thing is, now it is completely becoming a single cell. But inside this, more than one nucleus is present. So since many nuclei are present, now it will come under multinucleated. It will come under multinucleated. Understood or not? Already we studied about this in the cell chapter. This multinucleated, though it is multinucleated, how it is formed? Due to the fusion of the cells. Since it is formed due to the fusion, it is called syncytial. Okay, it is also called a syncytium. There is another word, that another word is called sinusotic type of multinucleated. But what is this sinusotic? This sinusotic will happen due to cell division. Suppose there is a single cell with single nucleus, and upon cell division it is forming the two nuclei, and after the nuclear division, after the karyokinesis, cytokinesis should occur. Suppose cytokinesis is not occurring, now what happens? In single cell, two nuclei are produced. That means due to failure of cytokinesis, after completion of karyokinesis, if multinucleate is formed, that type of multinucleated condition is called sinusotic. But if multinucleated condition is formed due to fusion of the cells, it is called a syncytium. In this way, multinucleated is of two types, syncytium and sinusotic. And here what we are saying, the skeletal muscle cells are examples for syncytium. That's why these are multinucleated cells. And generally nucleus is present just below the plasma membrane. Clear? And the plasma membrane of muscle cell is called sarcolemma. Yes or no? So anyhow, so all these names slowly we will study all these things. So this is what about the general differences or the comparison among these three. So you please understand this. In this diagram here, between the two cells, they are saying that junction between the adjacent cells, and this junction is called as intercalated disc. And it will help in quick movement of information from one cell to other cell, so that in the cardiac muscle cells, so if with respect to atria or with respect to ventricles, if you are observing, so they can contract at a time. To contract at a time, the information has to pass very quickly, then only they all can contract at a time. So in this way, for quick transfer, these intercalated discs or junctions are very important. Anyhow, that much was not given in this particular chapter, so we are not going into that particular part, so leave it. Now we are going with skeletal muscle. So please understand, in our syllabus we are talking only with respect to skeletal muscle. We are not talking about the smooth and cardiac muscle. So please observe carefully. And here they are talking about skeletal muscle. So please observe here. We are actually talking about four types of tissues. What are these four types of tissues? First one is epithelial tissue. Second one is connective tissue. Third one is muscular tissue. And the fourth one is neural tissue. So please understand. So that's why. So anyhow, first two we are not talking. So leave about this. And in the first two also, so anyhow, epithelial cells, many times we have studied about this. Under the connective tissue, in this chapter we will study about bone and cartilage. So please remember this. Bone and cartilage are examples under connective tissues. And now we are studying about muscular tissue. And in the next chapter, that is the neural control and coordination, there we will study about neural tissue. So these are all tissues. So please understand this. Suppose in the examination they are giving that muscle. The term muscle, please understand this. The term muscle is. So now they will give different options. It is a single cell, it is a tissue, it is an organ, it is an organ system. Likewise they may give. So if they are using a term called muscle, the term muscle represents tissue. It represents tissue. Many students are making mistake. That's why repeatedly I am focusing on this particular word. In the same way, the term nerve, N-E-R-V, nerve, is also a tissue. Nerve is a tissue. Please understand these basic words. Now now we are talking about the muscle here. Regarding the nerve, we can study in the different chapter. Regarding this muscle, if you are talking, Now it is tissue means what? Group of cells. Now what is that cell? The cell is called as a muscle cell. Please understand this. Simply if they are writing the word muscle, it is a tissue. If you want to refer the cell, then you need to write muscle cell. It has different names. Slowly I will tell you all the names, so don't worry. Now we are talking about the skeletal muscle specifically. Now since it is a tissue, it is having again group of cells. So now we will talk about this, and here under the cross section they are talking about this particular tissue. So in this muscle tissue actually, so these cells are having a particular pattern and they are forming some bundles, and again around these bundles some layers are present. So in this way in the NCERT they have given this particular section, so please observe this carefully. The same thing in a different diagram I am trying to represent, so please observe this carefully. Okay, so here what they are saying, this is completely, it is called as a muscle, and in this muscle again what happens, some bundles are there. Again there are some bundles are there, this is also one bundle, and again in each bundle again some cells are present. This is the single cell, this is the single cell, and this single cell is called as muscle cell. Muscle cell. What are the different names for muscle cell? Please remember, many students are not knowing these names, so that's why they are getting confused here. In simple, if you are saying, it is called as muscle cell. Muscle cell is also called as myocyte. Myo means muscle, cell means site. Please understand like this. Muscle cell is also called as myocyte. Both of them are same only. And how this muscle cell is present? The skeletal muscle cell, if you are observing, it is present as a long fiber. It is present as a long thread-like structure. So that's why we are calling it as a fiber-like structure. That's why it is called as a muscle fiber. All the names are same. You can say muscle cell, you can say myocyte, or you can say muscle fiber. So muscle fiber you are saying means you are talking with respect to single cell. It is a long thread-like fiber-like structure. So this is the muscle fiber or myocyte, single cell. And this is a single cell means the single cell is having plasma membrane. The plasma membrane of muscle cell is called sarcolemma. Lemma means membrane, sarco means muscle. It is the plasma membrane of muscle cell. That's why it is called as sarcolemma. This is single cell. Now inside this each cell again long thread like fibrils are present. They are called as myofibrils. Myo means muscle. They are fibrils. They are also fiber like, but they are very small. So they are small fiber like structures present inside a single cell. So in this way, inside a single muscle cell, again very long thread like structures called myofibrils are present. These are present parallel to the long axis of this particular muscle cell. Understood or not? That's why inside this myofibrils they are saying. So many myofibrils are present in single muscle cell called myocyte or myofibril. Like this is one muscle cell, one muscle cell, one muscle cell, one muscle cell, one muscle cell. Likewise, many muscle cells are grouped put together. That's why this is called as muscle bundle. Bundle. Please understand this, bundle. And this bundle is also called as a fascicle. In the NCERT, they have given a name called as a fascicle. What is this fascicle? This is nothing but a muscle bundle. It is the bundle of muscle cells. This is called one fascicle, one fascicle, one fascicle, one fascicle, one fascicle. Understood or not? In each fascicle again, muscle fiber, muscle cell or myocyte. This is one cell, one cell, one cell, one cell, one cell. Likewise, many cells are present. Understood or not? And inside each cell again, myofibrils are present. Understood or not? Try to understand this particular diagram. So in this way, now these are the bundles. Bundles are called as fascicles. Now many bundles will group together, and entirely it is called as one muscle tissue. Muscle tissue. Please understand this. Simply muscle. So in this way the names were given. At least in the NCERT, diagrammatically very clearly they have represented. So please understand all these things. Clear? So this is what the diagram is. So with respect to cells from inner side to outer side, let us come. So here it is the muscle cell or myocyte or muscle fiber. Many will group together and they will form muscle bundle or fascicle. Like many bundles will group together and form muscle. This is what the names that you need to understand. Now here we are studying another thing. That point is regarding the connective tissue. Around each cell, see, please remember here, all these cells are not directly attached to each other. In between the cells again, there is a connective tissue here. fibrous connective tissue is present. So in this way, now around each muscle cell there is a connective tissue layer that is called as endomysium. Around each cell endomysium is present. Now here this is the bundle called fascicle or muscle bundle. Around each bundle again connective tissue is present. That is called as perimysium, and in the NCERT they used a term called as fascia. Fascia. Fascicle is the muscle bundle. Around each fascicle, fascia is present. That is a connective tissue layer that is present. So around this one again, perimysium, you can also call it as fascia, is present here. Okay, and around the each muscle, complete muscle, there is a connective tissue layer that is called as epimysium. So outermost is epimysium, and around each bundle, perimysium. Around each cell, endomysium. So these are the connective tissue layers that are present. So better try to remember these names also. These names also. And if you remember these names, it will be easy to remember the names in the nervous system also. In the nervous system, what you will get? So here it is the muscle, there it will be the nerve. And here it is the fascicle, there also it is called as fascicle. Okay. And next one. And here it is the muscle cell, there it will be the nerve cell. Neuron will be there. So in this way you are getting. And with respect to layers, if you are talking, here it is the epimysium, there it will be the epineurium, mysium, muscle, neurium, neuron, right? It is epimysium here, there it will be epineurium. And here around this, around this bundle again, perimysium, but there it will be perineurium. And here it is the, around each cell it is the endomysium, there it will be endoneurium. In this way names are comparatively. Here mysium will come, there neurium will come. And similarly, you can compare this, you can easily remember this. So this is what about the structure with respect to the muscle cell. And here what we are saying, the plasma membrane of muscle cell is called as sarcolemma. Okay, please understand this. This is what the name that we are using here. And the cytoplasm is called as sarcoplasm. Okay, the cytoplasm is called as sarcoplasm. And the endoplasmic reticulum present inside muscle cell is called as sarcoplasmic reticulum. So all these names, simply they added a word called sarco. So in this way, with respect to muscle cell, they are using this. That sarcoplasmic reticulum, that is nothing but endoplasmic reticulum of muscle cell, that is actually helping in storing of more and more calcium ions, and that calcium is useful for muscle contraction. So please remember this. So this is what about the general information regarding this particular diagram. With that, some basic information is completed. Then in the NCERT, actually they have given about this particular structure. But before going with this structure, first we need to understand the structure of contractile proteins. Then it will be very easy to understand the next content, and then we can directly go with contraction. Okay. Structure of the contractile proteins. Here actually they are talking about two types of names. What are these names? One is called as a thin filament. One is called as thin filament, and the other one is called as thick filament. Thin and thick filaments. Okay. Next one is thick filament. Thin and thick filament. Now, what are the components that are present in the thin filament? This is very important. Thin filament is having actin, tropomyosin, and troponin. Please remember here, this is the thin filament. This thin filament has actin, tropomyosin, and troponin. I will tell you one after the other. And whereas the thick filament is having myosin. So please observe this very carefully, because these are the names that we are using under the mechanism and all. So here thin filament, thick filaments are present. Thin filament is having actin, tropomyosin, and troponin, where the thick filament is having myosin. Is it clear? Now, how this actin is formed and how it is present. So in this way, for each one we will study one by one. Now let us observe the NCERT diagram. This is the NCERT diagram. So here they have given the labeling as F actin. What is this F? F means filamentous or filament. So why it is called as a filament? Filament means what? It is a thread-like structure. So these round, you just observe carefully. This is a long structure. It is a long thread-like structure. That's why they are using a term called as a filamentous. Understood or not? Here, how many filaments are present here? See this one. This is one and this is the other one. So in this way, two long filamentous actins are present. They are helically coiled with each other. In this way, they are present. and since they are long thread like, they are called as F-actin. Very good. And how this F-actin is originally synthesized? In the F-actin, again, if you are clearly observing, there are round structures. Round means they are globe-like. So that's why it is called globular actin. So please observe carefully. Here they are using some terms. One is called as G-actin. G for globular. It is a globular. It is a globe-like. Globe means it is a spherical shaped. So in this way, many spherical shaped G-actins, they will attach to each other. Individually, each one is called as G-actin. When many are attached to each other, they are making a long filamentous structure. That's why it is called as F-actin. So in this way, F-actin is a polymer of G-actin. This is what they are saying. In this way, two filamentous structures, helically they are coiled with each other. In this way, F-actin is present. That's it. With this, information regarding F-actin is completed. Next one. Now we will talk about the tropomyosin. And this tropomyosin, this is also filamentous structure. And how many? Two filamentous structures. These are also helically coiled. And helically coiled, and they are interacted with this F-actin. This is what the information regarding tropomyosin. Over. Next we are talking about the troponin. Please remember, troponin is not a continuous filamentous structure here. Many troponins, now they are present here and there. And where they are present? Technically, if you are observing this troponin, observe the diagram here. Three small round structures they have given. They are actually the subunits. In each troponin, three subunits are present. And you need to understand what are these three subunits. Very clearly not they have given in NCERT, but you need to understand this. Troponin, it is represented with Tn. It is the troponin. And it has three subunits. Troponin has three subunits. Troponin has three subunits. What are these three subunits? One is troponin C, one is troponin T, one is troponin I. C, T, I. You need to understand the meanings of this. You can easily understand this. So what this Tn means troponin, you can understand. What is this subunit troponin C will do? C, it will bind to calcium. It binds to calcium. During muscle contraction, calcium ions are required. And where these calcium ions are stored? Just now we studied. Calcium ions are stored in sarcoplasmic reticulum. If there is a nervous signal, there is a signal from nervous system for the release of this calcium for the muscle contraction, then what happens? That information is coming till this muscle cell, and inside this muscle cell what happens? The information is keep on moving, and from sarcoplasmic reticulum calcium is released into cytoplasm. Now in this cytoplasm, these myofibrils are present. Yes or no? In these myofibrils, these thick and thin filaments are present. And in the thin filaments, this troponin is present. In the troponin, this troponin C, the calcium binding subunit is present. Now this calcium will bind to this particular subunit. So calcium will bind to this troponin C. Understood or not? So in this way, it is having the role. Now next one. Troponin T means it is tropomyosin. It binds to tropomyosin. It binds to tropomyosin. Observe the NCERT diagram carefully. Where they have attached this. So one subunit is attached to tropomyosin. Observe carefully. In this way, one subunit is attached to this thin filament through this tropomyosin. In this way, it is becoming part of thin filament. Clear? And there is a third subunit. And that third subunit is I. I means it is inhibitory. It is inhibitory. What it is inhibiting? Now you need to understand this mechanism. If you understand the function of this inhibitory C, anyhow this is normal. This is helping in binding to tropomyosin, that's it. Now these two are very important. If you understand the functions of these two subunits, then only you can understand the mechanism of muscle contraction. So since these two are important to understand muscle contraction mechanism, deliberately I am talking about these two things. Clear? Now I will draw one rough diagram, try to understand the mechanism, so it will be easy for you. Now what we will do is, suppose this is the thin filament. Please remember I am using the term called as thin filament. Thin filament means what are present? Actin is present, tropomyosin is present and troponin is present. Is it clear? This is how thin filament is there. And now we will talk about the myosin, thick filament. I will tell you anyhow, so don't worry. For the myosin, what happens is it is having some head-like structures like this. There are two head-like structures, so please observe carefully. Now they have to, this is the part of myosin. Each subunit is called meromyosin. I will tell you this space separately. For this meromyosin, if you see here, it is having some head-like structures. And for these head-like structures, here it is having actin binding sites. Actin binding sites. Actin binding sites. And what actually it is? It is part of myosin. Myosin. Please observe carefully. Myosin is trying to bind to actin. Myosin is trying to bind to actin. So here for the on the actin, this has to bind. So that's why to bind to the actin, myosin is having actin binding sites. So in this way, if these two are free, now they can attach to each other. But please remember, if it is attaching means when it will attach, you need to understand this under the mechanism. Anyhow, the information is interlinked, so that's why here itself I am covering that. When it will attach, it should attach. Myosin should attach to actin only during muscle contraction. Please understand this. When this myosin should attach? Only during muscle contraction. That means when there is no information for muscle contraction, that means in the relaxed state, it should not, myosin should not attach to actin. Myosin should not attach to actin means someone should come in between this. Now understand this carefully. Myosin should not attach to actin means someone should come in between this. Now who is this coming in between? It is the troponin I. It is the troponin I. This is what we are studying here. What is the function of this troponin I? It is inhibitory. What it is inhibiting? It is inhibiting the binding of myosin with actin. How it is binding? How it is inhibiting? By present in between these two. Understood or not? Above is the actin binding, actin, and below is the myosin, and in between this inhibitory is there. Between these two, inhibitory is present. Understood or not? Since inhibitory is present in between, now these two cannot bind. So please understand the conclusion here. During the resting stage, that means when there is no information for muscle contraction, at that time this I inhibitory is present between actin and myosin, so that these two are not interacting. Since it is inhibiting the interaction of actin and myosin, this subunit is called as troponin I, inhibitory subunit of troponin. That's it. So these are the three subunits and these are the functions. Anyhow, this is helping in binding to tropomyosin, but these two are important. Please understand here. When there is no information for muscle contraction, then this I is present between actin and myosin, so that it is inhibiting the interaction between these two. And what this calcium will do? If there is an information for muscle contraction, now the information is coming to the muscle cell. In the muscle cell, what happens? Calcium will be released from sarcoplasm into the cytoplasm. Sarcoplasmic reticulum into cytoplasm means that is the sarcoplasm. Now the calcium present in the cytoplasm, it will come and bind to this. When it is binding to this troponin C, then what happens? Binding of calcium to troponin C will cause removal of I from that position. When I is removed, what happens? Now these two can attach, contraction will occur. Anyhow, under the contraction, once again very clearly I will explain, so don't worry. Here you just understand simply the meanings and the functions of these particular subunits. That's it. So with this we have completed the thin filament. Thin filament is completed. Now we need to talk about the thick filament. What is the thick filament? Thick filament is the myosin. So many units. What is the each unit is called as meromyosin. Please understand this. Each unit is called as meromyosin. Meromyosin is the monomer. One unit is called as meromyosin. Likewise, many meromyosins will attach and then they will form the myosin. So that's why in general, if you are talking about the thick filament, they will give like this. This is the thick filament, suppose. And for this thick filament, if you are clearly observing the diagram, they will give something like this. They will give like this. Observe carefully. Observe the direction here. I am drawing it towards this, towards like this. And suppose this is the center. And here they are positioned like this. You need to understand this direction also. Some students what they are doing is, suppose this is the myosin, they are thinking that always it is like this. No, it is not like this. It is not like this. Please observe carefully. So this side they are like this, so that they can pull the thin filament in this direction. They are in this direction so that they can pull the thin filament in this direction. So please observe these things very carefully. Here observe carefully. Here, this is the diagram. This is the center. Now they are present on this side. These are present on this side. So now what is the advantage of this? Now they will attach to this thin filament and they will pull it towards the center of a band. And this side they are pulling it towards center of a band. So please observe these directions carefully. Many students are not noticing these directions. They are thinking that all of them are in one direction only. No. Now these are towards center. This side are also towards the center of a band. So it means center of the thick filament. Please observe carefully. Don't make mistakes. These are some small mistakes that you are doing. So ultimately you are thinking the concept in a different way. So don't do these mistakes. Okay. Now we are talking about one unit. So in this way, many things they have shown. We are talking about one. That one is called as meromyosin. And here also some students are making mistake. The students are thinking that this one is one meromyosin, this one is another meromyosin. This is what they are thinking. This is wrong. This is wrong once again. Please observe carefully. These two combinedly is called as one meromyosin. These two combinedly it is called as meromyosin. This is the diagram. So observe very carefully. So two are present here. Two are present. Please observe carefully. So that's why you need to study two together. So two together is called as one meromyosin. So this is the basic unit. And in this way many will combine together and they are forming the myosin. So that's why you are studying like this. So that's why if they are asking that in one meromyosin, how many ATP binding sites are present? This is one ATP binding site, this is one ATP binding site. Combinedly it is one meromyosin. So one meromyosin has two ATP binding sites and it has two actin binding sites. So this is the original logical content. Don't make any mistakes. Don't make any wrong interpretations on your own. Please do observe this very carefully. So now this is completely called as a meromyosin. In this meromyosin, now this portion is called as a tail portion. And this tail portion, only this tail portion is also called as light meromyosin. Only tail portion is called as light meromyosin. And now from this main line, if you are observing, there is a portion that is projected outside. And this projected outside is also called as a cross arm. This is also called as cross arm. And for this cross arm, at the top, it is having some round head-like structure. This is called as head. So in this way, technically, if you are observing, it is having three portions: head, cross arm, and then tail are present. Only tail is called as light meromyosin, whereas head plus cross arm, this part is called as heavy meromyosin. This information was very clearly mentioned in NCERT. So please observe the NCERT content carefully. So heavy meromyosin has head plus cross arm. Light meromyosin is only the tail portion. Clear? Now in this way, many will combine together and they are making myosin, that is also called as thick filament. Clear? This is what the general information is. Now on this head portion, now it has ATP binding sites. Now ATP will bind here. Not just binding. Once ATP is bound here, then what happens? Hydrolysis of ATP will occur. For the hydrolysis of ATP, ATPase activity is required. So this is having ATPase activity also. That's why when ATP is bound to this, it is hydrolyzed to ADP plus PI. It will be hydrolyzed to ADP plus PI. So please remember this information in this way. And then just now I told no, myosin is binding to actin. Myosin is binding to actin. So this is what our diagram is. Myosin is binding to actin. So that's why it is having actin binding sites, so that it will bind to actin. But under the resting condition, in between these two, who is coming? Troponin I is coming. That's why these two cannot interact. But when calcium is attached here, when calcium is attached to troponin C, to this calcium is attached. When this is attached, what happens? Now this troponin I will be released. This troponin I will be released. Once troponin I is released, now what happens? Now there is no inhibition here. Now myosin can bind with actin. So that now it will help in pulling it towards the center of myosin, thick filament. Towards the center of thick filament, the pulling will occur. So in this way, this mechanism will occur, and anyhow I will tell you this under the mechanism. This is what about the structure of these contractile proteins. At least NCERT information you please try to understand. Don't try to by heart this. Understand this. This is what they are saying here. Okay, this is what about the thick and thin filaments. Next we are talking about the structure, functional unit. The functional unit of the skeletal muscle is called as sarcomere. The functional unit is called, each unit is called as sarcomere. What is this unit? Now here from this Z line to the next immediate Z line. So the portion between the two successive Z lines is called as sarcomere, and this is the functional unit. This is the functional contracting unit. And what are present in this? How exactly it is present? So now we will observe this very carefully. Anyhow, the NCERT diagram is very clear. Now observe very carefully here. Now actually this Z line is actually a membrane. Z line is a membrane. This is also a Z line, that is also a membrane. And attached to this Z line, what are present? Observe the diagram carefully. Attached to this, what are present? Thin filament, thin filament, thin filament, thin filament, thin filament, thin filament, thin filament. That's it. That means to this Z line, actually it is a membrane. To this, what are attached? Only thin filaments are attached. And this dark red is the thick filament. And this thick filament is not attached to Z line. So observe the diagram carefully. Only thin filaments are attached to the Z line. Here also, to the Z line, only thin filaments are attached. Thick filaments are not attached to Z line. So please observe this very carefully. And here let us consider that this is a resting stage. This is the resting stage. Resting stage means what? It is muscle is not undergoing contraction. The cell is not undergoing contraction. Clear? And now what happens? Now here at the center, here it is having the thick filament. What is this thick filament? It is the myosin. It is the myosin thick filament. Already I told you, now these directions are very important, because while they are pulling it towards the center of thick filament. Understood or not? In this direction they are present, and this thick filament is not attached to this Z lines or Z membrane. Okay, clear? Now this is the thick filament. Under the resting state, how exactly it is present? So some portion of thick and thin filaments are overlapped with each other. Only some portions. And what are these portions? Generally the ends of thick filaments. Ends of the thick filaments, they are actually overlapped with thin filaments. So that the central portion of this thick filament is not overlapped with the thin filaments. Understood or not? The portion of the thick filaments that is not overlapped with thin filaments is called as H zone, Hansen's zone, H zone. And at the center of this H zone, even though here they have not mentioned, at the center of this H zone there are some proteins. They are present as a membrane, but it is not like a plasma membrane. There are some proteins, they are actually present at the center, so that they are helping in keeping the thick filaments in that location. Please remember, in the muscle contraction process, thick filaments will never move. Thick filaments will never change their locations. They are present in that location only. How that location is maintained? Because at the center there are some proteins, they are making one M line. There is an M line at the center, and the M line is nothing but some proteins, and they are helping in maintaining the thick filaments in that position. So that's why please understand, in the process of contraction, thick filaments are present in that location only. And who will move? Thin filaments will move. Thin filaments will move. Towards which side? Towards the center of this thick filament they are moving. Is it clear? So in this way, now these are the thick filaments, and the ends of the thick filaments are overlapped with the thin filaments, but at the central portion, now under the resting stage, it is not overlapped with thin filaments, and that particular region is called as H zone. And at the center of the H zone there is a protein membrane. That protein line is called as M line. This is the normal condition. And now we will talk about A band and I band. A means anisotropic band, whereas I means isotropic band. So now you need to observe that what are actually present in the I band. What is this I band? Observe carefully. This is the thick filament. For the next one again, it is the thick filament. So between two thick filaments, between two side by side thick filaments, there is a portion. There is a portion, and this portion is called as I band, isotropic band. So what exactly are present in this I band? I band is having the Z line. I band is having the Z line, and along with the Z line, it is also having the thin filaments. Here what are present? Z line and thin filaments are present. That's it. But I band is not having thick filaments. Very important for examination. So please observe carefully. I band is having Z line and thin filaments. That's it. Understood or not? Now come back to this A band. What is this A band? Between two I bands is the A band. And here in the A band, how exactly we are deciding the length of A band? Please observe carefully. During muscle contraction, A band length will not change, but I band length will decrease. Why? Why you are saying like this? Because while you are defining the A band, then itself, you are saying that A band is actually the length of this particular myosin. Whatever is the length of this myosin is there, only that much is the A band. And just now I told you, during muscle contraction, myosin is not undergoing contraction. Myosin length will remain same. Since myosin length remains same, A band length also remains same during muscle contraction. This is the logical point. Since when we are deciding this A band, then we are saying that it is actually the length of the myosin. But technically, if you are observing, A band is not just having myosin. It is not only having myosin. Along with myosin, even it is having thin filaments. So that's why what you need to observe. A band is having both thick filaments and thin filaments. A band is having both thick and thin filaments. But I band is having only thin filaments. It is not having thick filaments. Observe it very carefully. Clear? This is what the general arrangement of this particular one. And here from one Z line to the next immediate Z line, this particular portion is called as sarcomere. So that's why generally when they are talking about the sarcomere, so they may ask you to identify the correct formula for this sarcomere. If you are writing this sarcomere, how we can write this sarcomere? One sarcomere is equal to, this is the sarcomere. In this sarcomere, what is present? This is completely A band. That means one complete A band. This is one A band. One A band plus what is there? Now, what is this portion? Now, this is actually not complete I band. This is complete I band, right? Up to this one, what is it called as? It is half of the I band. This is half of the I band on this side, and this is also half of the I band on this side. So that's why what is one sarcomere? One sarcomere is equal to one complete A band plus two half I bands. Two half I bands plus two half I bands. Please understand this very carefully. This is the only way you need to write. So one complete sarcomere is one complete A band plus two half I bands. Two half I bands. This is how only you need to write. But some students, what they are doing, since they are very good at mathematics, what they are doing, they are writing that. So please remember, whatever I am writing now, this will be wrong. Okay. So sarcomere is equal to A band plus 2 into half I bands. 2 into half I bands. Now what they are doing? 2 cancel. So now they are writing that it is A band plus 1 I band. This is what they are writing. Since they are very good at mathematics, they are doing this simplification and they are ultimately writing that this is. But this is a blunder mistake. What is the meaning of A band plus I band? Observe carefully. What is the meaning of A band plus I band? A band means it is starting here and I band means it is going here. Now from this to this, is it called a sarcomere? No, it is not sarcomere. So that's why sarcomere is one complete A band plus half plus half. So two half I bands. This is what is called as sarcomere. But if you are writing that sarcomere is equal to A band plus I band, that is wrong. That is wrong. So please don't apply your mathematics in this particular case. This is a wrong concept. You should not do like this. it is always like this only. It is like this only. So sometimes they may give like this also. So you need to understand everything is from NCERT only. I am not even going is even a single point out of NCERT. So please observe it very carefully. Okay, this is what about the sarcomere, A band, I band, and everything. Clear? And with this, the basic arrangement, and where we are actually observing this type of arrangement. This arrangement is actually observed in the myofibrils. Inside the muscle cell, myofibrils are present, no? So in the myofibrils, we are observing all this. In the myofibrils, we are observing all this. And whatever is A band is there, that is the dark one. And whatever is the I band is there, this is the light band. And alternatively, dark light, dark light, dark light are present. That's why we are observing the striations or stripes. Stripes, yes or no? This is what it is called as striped appearance or striated appearance. Understood or not? This is what they are saying. And you can see this. This is the actually A band. Even in the A band also at the center, a light area is there. And what is this light area is called as H zone. And in this H zone again at the center there is a thick line. And what is that line will be called as? M line. It is called as M line. So this is what the NCERT diagram. So please observe it very carefully. Clear? And now we are going with mechanism of muscle contraction. And this mechanism of muscle contraction is explained with the help of sliding filament theory. Sliding filament theory. This is the actin, this is the myosin. These two are interacting because myosin has actin binding sites. Now these two are interacting. But here, along with the actin, what are present? Tropomyosin is present, troponin is present. And generally what happens? Under the normal conditions, what happens? Troponin I is coming in between. That's why it is inhibiting the interaction. But when calcium is attached to troponin C, now what happens? This I will be released. Now it can interact and now it can help in pulling of this. And please understand, once again I am telling you, who is pulling? Myosin is pulling. And whom it is pulling? And it is pulling the light filament. Technically it is the actin, that means entire thin filament is coming here. So thick filament will be here itself. Thin filament is coming like this. Originally it is here. Now because of the pulling, what happens? It is coming towards the center from this side. And what about this side? It is pulled towards this side. That's what I am saying, that from the both the sides now it is pulled to the center of A band. This is what the point that you need to remember. Now slowly I will explain this. I will explain this information, and then in the NCERT they have given this particular diagram also. In the normal relaxed state, what they are saying, this is the thick filament, thin filament. Thin filaments are present at the ends of the myosin. At the center there is a clear cut space, and this is called as H zone is present. Understood or not? And here also I band is having this much space. At the center Z line is there, attached to this thin filaments are present. This is what in the normal relaxed state. And when it will be undergoing the contraction, and what happens during the contraction, now observe carefully. And from where actually this contraction will begin. This point you need to understand. In the NCERT, very clearly they have given the sentences. In short, I will try to tell you this. And here we are talking about the muscle contraction. And these muscles whatever we are studying are skeletal muscle. Now you need to understand this point here. Skeletal muscle and smooth muscles. For the contraction of these muscles, information should come from the nervous system. Please observe carefully. For the contraction of skeletal and smooth muscle means visceral muscles, the information for contraction should come from the nervous system. Then only they will contract. Understood or not? But what about the cardiac muscles? Cardiac muscles present within the heart. Already in the body fluids and circulation we studied. Cardiac muscles are auto-excitatory, self-excitatory. Already I told you this. There is a SA node, AV node, and they are controlling their own contractions. Yes or no? That is not depending on the nervous system. Even nervous system, sympathetic, parasympathetic nerves, they can control. But originally, cardiac muscles are not under the control of nervous system. They can excite on their own. They can contract on their own. So this point you need to remember. And here what we are studying? We are studying for skeletal muscle. So skeletal muscles, they cannot contract on their own. These points are very, very important. They can interlink these two concepts. So please observe carefully. Skeletal muscles cannot contract on their own. So that's why compulsorily the information should come from nervous system. Understood? So from the nervous system, now the information is coming. How it is coming? Through the neurons it is coming. So it is initially passing through several neurons. Ultimately, the last neuron is connected to the cells of the muscle. and that will form the junction that is called as a neuromuscular junction. Please understand this. Ultimately, the last neuron will be connected to plasma membrane of muscle cell. So there the junction is called as neuromuscular junction, or it is also called as a neuromotor junction. Okay, so it is also called as a motor end plate, because that nerve that is coming is called a motor nerve. Okay, and the motor neuron is will be there, and the motor neuron is ending there, so it is the motor end plate, and that is actually the neuromuscular junction. Clear? So in this way, information for the contraction is coming from the nervous system. Finally it reached the muscle cell. Now what happens? Now the information, so that excitability concept you just understand. Now the muscle cell is undergoing excitation. Some potential changes are taking place inside the muscle cell also. And because of these potential changes, ultimately what happens? Inside the muscle cell, now you need to understand this point. I will draw one rough diagram, so observe carefully. So since it is a cylindrical structure, so I am drawing like a cylinder, observe carefully. Skeletal muscles are cylindrical in shape, right? Now here somewhere, let us talk about some endoplasmic reticulum. This is called as sarcoplasmic reticulum. Sarcoplasmic reticulum. Sarcoplasmic reticulum. Simply I am drawing some rough diagrams, so observe carefully. Suppose this is the thin filament, okay, and then this is the thick filament. This is the thick filament. Let us consider like this. Anyhow, these thin filaments are attached to Z line like this. Just now we have studied, no? So they are present like this. Like this they are present. Nice. And on this thin filament, just now we studied, no? Even tropomyosin is present here. Only for one I am drawing. I am not drawing for everyone, everything. So please observe carefully. On this tropomyosin, what are present? There are actually 3 subunits, right? 1, 2 and 3 subunits are present. Observe carefully this point. Now initially it is in relaxed state. When it is initially in relaxed state means there is no information for muscle contraction. It is in relaxed state. Now what you are saying? Now we are saying that the calcium is stored inside the sarcoplasmic reticulum. And here this is the troponin I. It is present between actin and myosin. That's why now these two cannot interact. These two cannot interact. For this one also. Now let us draw this. This is how. Now it is not directly attached to the actin. Now in between this troponin I is there. Now it is inhibiting the interaction between these two. This is the relaxed state. This is the relaxed state. Now we will talk about the neuron. Now the neuron at the through that axon, right? Last axon. Now this is connected to this particular plasma membrane of muscle cell. It is also called as sarcolemma. Sarcolemma. Now the information from the neuron is coming, and now it ultimately came to the muscle cell. And now what happens? Now across this muscle cell again, potential change will occur. So that now this muscle cell is undergoing excitation, excitability property. And because of this, ultimately what happens? Calcium stored in sarcoplasmic reticulum will be released into the sarcoplasm. Sarcoplasm is what? Cytoplasm of muscle cell. Released into the sarcoplasm. Now what this calcium will do? This calcium will go and bind to, this is troponin C, calcium binding. So now this calcium will go and bind to troponin C of troponin. And now what happens? If calcium is attached to this, now what happens? There is a structural change, so that now this inhibitory part will come outside. Now this inhibitory part will come outside. Now observe this carefully. This is the myosin like this, and now this is the actin, and this is the tropomyosin, and now troponin. Already calcium is attached here, Ca+2. So that's why what happened? Now its position got changed. Initially it is present in between this. Now it is coming outside. Once it is coming outside, what happens? Actin and myosin can directly interact. Suppose this is the actin, this is the myosin, and in between this is the inhibitory. But when calcium is binding to troponin C, now what happens? Binding of calcium will cause change in the structure, so that inhibitory is released. Once inhibitory is released, what happens? Myosin can bind to this. So that now it will bind here and it will start the muscle contraction. And please remember, this side is pulled in this direction and this side is pulled in this direction. So in this way, it is coming to the center of a band. So that's why what happens now, these are pulled, but the myosin thickness, myosin length will remain same. So that's why a band will remain same. But these thin filaments are pulled. Thin filaments are pulled means this Z line is also pulled. So in this way, what happens? The length of this particular I band will decrease, but this A band will remain the same. Anyhow, slowly one after the other we will understand, and this is what ultimately that is happening. I hope now you understood what is the role of this calcium release and what is the role of troponin. Troponin will play a very significant role that you need to understand. In this way, initiation will occur. So now you need to understand that inhibitory part is removed. Now myosin can bind with this actin. And now what happens? Now how exactly this cycle is taking place? Don't think that the same is keep on pulling up to here. Some students, what they are thinking is, now it will bind here, it is only bringing up to here. No, that is not the right concept. Now what happens? Now observe carefully. Suppose on this side I will tell you, because I can keep the hand like this on this side. Now what happens? Myosin is binding here. Some students are thinking, I am telling you the wrong one, so observe carefully. Some students are thinking that the same thing, whatever is binding here, it is pulling up to here. No, it is not like this. This pulling of thin filament is a sequential process. First it will bind here, pull something, and leave it, and bind here, pull, and leave, bind here. So leave, bind, attach, pull, again leave, attach, pull. In this way, slowly, gradually it is pulling. When it is gradually pulling, now there should be a cycle. What is this cycle? For attachment to actin, and then pulling it, and then releasing, and once again freshly attaching it at different location, further pulling and leaving, and freshly attaching, pulling. So this is the cycle, and this cycle is very simple one. And for one complete cycle, for one cycle it is 1 ATP. Once again I am telling you this point. For one complete cycle, 1 ATP is required. Please understand this. Some teachers are saying that 2 ATP are required. That is wrong concept. For one complete cycle, 1 ATP is required. Anyhow, now I will tell you, right? So now observe carefully. And from where we need to start this cycle? For easy understanding process, what we are doing is attachment of ATP so that cross bridge is broken. Now understand this. Now observe this carefully. Okay, many students are getting so many doubts. Even though 99% of the concept is clear, those 1 or 2 points you are making a mistake, and there is a chance that they may ask that point in the examination. So observe very carefully. Now here what they are saying is, suppose this is the actin. We are directly focusing on the actin and myosin. Okay, and this is the myosin. Okay, myosin like this. It is pulling in this direction. Okay, the movement will be in this direction. Observe carefully. So it is attached. It is attached. But please observe the NCERT diagram carefully. This is the myosin that is attached to the actin. And this ADP and PA got released. Observe carefully. ADP and PA got released. And here in this particular structure, ADP, PI, ATP, nothing is present here. Observe carefully. ATP is anyhow not present. ADP and PA also got released. That means here none of these three are present. And when none of these three are present, then what is the final condition? To the actin, myosin is attached, and it is present like this. And now here ATP is not there, ADP is not there, PA is not there, nothing is not there. Now it is attached, and it is in this position only. And when this will be released, when this cross bridge will be broken, very clearly they mentioned in NCERT, observe carefully. When this cross bridge will be broken, when a fresh ATP binds, please remember here. Now what I will do is, now I will do like this and like this. Please understand, I will try to rotate this hand. What is the meaning of this rotation? Here my meaning of this rotation is change in the structure. Now observe carefully. When ADP and PA are released, now it is having a particular structure. Understood or not? And now what happens? Now it is attached like this. And now when it comes to here, what happens? A fresh ATP will bind here. A fresh ATP binding of ATP will change the structure. Please understand these concepts. Initially it is like this. A fresh ATP is bound here. When fresh ATP is binding here, Binding of ATP will cause the change in the structure. So now it cannot bind here. So now it will cause the release of this particular cross bridge. So cross bridge is broken. So initially it attached here, right? Now it is released. After release, what happens? Now here ATP is there. Now this ATP will undergo breakdown. When ATP is broken, then what happens? ADP plus PA are formed. But once ATP is broken, what happens? Structure will again change. Understood or not? And but this time, initially it attached here, but this time it will attach in a different position. So please understand here, attachment of ATP is causing change in the structure. And breakdown of ATP into ADP plus PA will cause again regaining the structure, so that it will attach at a different location. It is attaching at a different location. And then what happens? And along with this ADP plus PA, some energy is released, right? And using that energy, it will pull it. Pulling is towards which direction? This pulling is towards center of this pulling is towards this center. So these two are pulled in these directions. Observe it very carefully. This is the mistake done by many students. So in this way, it is pulling it to the center of that H zone. It is pulling in this direction. And after pulling, what happens? With the help of energy, it has pulled it. And now what happens? Again, this ADP and PA are released, but still it is attached like this only. ADP and PA are released, but still it is attached. When fresh ATP binds, then again it will change the structure. And then this ATP again, it is broken down to ADP plus PA, so it will attach in a different position. Using the energy, it will pull it, and ADP and PA are released, and once again fresh. So in this way, once ATP is bound in this cycle, it is coming up to here, and fresh ATP will bind, and again one new cycle. Please understand this. So in this way, for one cycle, one ATP is required. Calculation, if you are doing, 1 ATP per cycle is required. This is what happening is. Very simple mechanism. Attachment of ATP is causing change in the shape of this particular structure. So it is separated. So attachment, when ATP is attached, then only it will be detached from this. Then only it will be separated. Cross bridge will be broken. Or else still it is present like this only. That's why at the end of the paragraph, or generally we are saying that there is an example here. When an individual is dead, if they die, if someone is dying, now what happens? After the death, please understand, after the death what happens? ATP, further ATP synthesis does not occur. So that's why for the dead person what happens? Fresh ATP is not there. Suppose they are holding, so generally in some movies they will show that. So when an individual is dying, suppose they are holding a paper and they are dying. Now that paper will be like this. And to open these fingers, you need to use so much of energy, because these are locked here. Why they are locked? Please understand, this is a contraction. So in this contracted state, now they are present like this. And since individual is dead, there is no fresh ATP. If ATP is not attached, now it will not open. So that is the reason why now it is held like this. Please understand this. And in this way, it will be present like this. After few hours, what happens? Since the individual is dead, breakdown of these particular components will occur. Once breakdown of these proteins occur, so automatically everything will become loosened. Yes or no? This is what the question, application question, that they may link here. So that's why please understand, attachment of fresh ATP causes separation of the cross bridge. Understood or not? And that too, attachment of ATP is causing the change in the shape, so that it is separated. And upon breakdown, what happens? ADP plus PA is formed. So that's why again it will get its original shape, and it is binding at a different location. And now it is causing the pulling with the help of the energy. And once it is pulling, what happens? Now this ADP and PA are released, and again a fresh ATP will bind, and it will be again separated, and it will bind, and it is a continuous process. So this is what about the sliding filament theory. So in this way, who is sliding over whom? Myosin is present there itself. Myosin is present there itself. Only these thin filaments are getting slided. So that's why this A band, myosin, the length of myosin will remain same. So that's why A band size will remain the same. But who is moving here? Thin filaments are moving. Since thin filaments are overlapping more, during the relaxed state, overlapping is very less. But during the contraction, observe carefully. During the contraction, more overlapping is observed here. This is what. So because of this pulling, what happens? Now these are coming very close, so that I band size is decreasing. So now during this muscle contraction, what happens? A band size remains same, but I band size decreases. I band size decreases. And next point. So during the relaxed state, H zone size is more, but during contraction, H zone size is decreasing, and in the maximum contracted state, H zone may completely disappear also. It may disappear also. These are the some important points that you need to understand from this muscle contraction. So please observe this very carefully. And here what they are saying, suppose if you are doing continuous muscle contraction, suppose I am running for continuously, after some time what happens? I will start getting the pain in these calf muscles and all. Yes or no? Why I am getting the pain? That pain is due to accumulation of some acid. What is that acid? Lactic acid. The thing is, why lactic acid is produced here? In the normal conditions, the thing is, whatever the oxygen that we are taking from the air, it is going to the lungs. From the lungs, the oxygen will diffuse into the blood. And now this blood will be carried by mostly it is carried by the hemoglobin. Now it is supplying to all the tissues. So that even muscle cells are also getting the blood. That means even muscle cells are also getting the oxygen. So using the oxygen under the aerobic pathway, they are producing more and more ATP. This is the normal condition happening. But when I am continuously running for longer duration, what happens? The amount of oxygen that is supplied to the muscles, it is getting used immediately. So now there is a requirement of more and more oxygen. But that much of oxygen is not supplied to the cells, muscle cells. When enough oxygen is not supplied to the muscle cells, then what happens? Now you can see some anaerobic conditions in the muscle cells. When anaerobic conditions are formed, please understand, muscle cells have to run that particular process. So they have to continuously produce the ATP. So to continuously produce the ATP, but oxygen is not there. So to continuously produce the ATP, it is depending on anaerobic process. And here in the anaerobic process, what happens? Anyhow, glycolysis up to the formation of pyruvic acid is a common process. But now this pyruvic acid, 3-carbon pyruvic acid, is converted into 3-carbon lactic acid. So CO2 is not released here, but lactic acid is produced. So pH values keep on decreasing, and now it will start showing its effect on the muscle cell. So that's why we are getting the pain. And now we can call this as fatigueness of the muscle or muscle fatigueness. So in this way, accumulation of the lactic acid due to anaerobic breakdown of this glycogen will break down and form the glucose. That glucose will undergo glycolysis and form the pyruvic acid, and that pyruvic acid upon aerobic condition, it is forming the lactic acid, and this lactic acid accumulation is causing the muscle fatigueness. That's it. Understood or not? This is what the overall mechanism that is happening. So in that case, what we need to do? Better take the rest and then give the glucose and also give the enough oxygen, take the breath. So depending on the amount of the lactic acid that is produced, so it will take some time to become normal. That's it. So slowly this lactic acid from that muscle, now it will go to the liver, and there Cori cycle will happen, and now it will be used for some other process. But it will take some time, so until then we need to bear the pain. That's it. Clear? This is what they are saying here. And in the NCRT, next they are talking about some red muscles and white muscles. So what are these red muscle fibers and white muscle fibers? In the name itself it is there, so directly we can go with some few important points. Red. Why it is red? Because these muscle fibers, they are having a protein called as myoglobin. Already I told about this myoglobin in biomolecules, because it is a single protein molecule. It is a single protein molecule. So highest conformation is tertiary. I told about this. And regarding the myoglobin, when we studied about breathing and exchange of gases, under the oxygen dissociation curve, even in the revision lecture also I told about this. Under the oxygen dissociation curve, I told about this. Yes or no? It is showing that hyperbolic curve instead of sigmoidal curve. Reasons, everything we studied, that is the myoglobin. So this myoglobin is an oxygen binding protein, and these red muscle fibers, they are having more and more myoglobin, so they can trap more and more oxygen. So whenever it is required, they can give more and more oxygen. So that's why in the red muscle fibers, generally what happens? Aerobic process is taking place. Aerobic breakdown of glucose is taking place. That's why more and more ATP energy is produced. Yes or no? And that too, these cells are having more and more mitochondria. So that's why that oxygen will be utilized, and in the mitochondria, that Krebs cycle, electron transport chain will occur. So more and more ATP is produced. And these are the some informations related to these red fibers. But there are some muscle fibers, they are called as white muscle fibers, because the amount of myoglobin is almost negligible, very less. Okay, so that's why what happens, and here oxygen availability is very less in these cases, and that too the amount of mitochondria are also very less in these cases. So that's why in majority of the cases these cells are depending on anaerobic process for obtaining the energy. But here there is one advantage. The advantage is, even though ATP energy is produced is very less, the sarcoplasmic reticulum is extensively developed in these white fibers. Since sarcoplasmic reticulum is extensively developed, now more calcium is available here, and this calcium release can cause strong contractions. So that's why overall conclusion, if you see, even though white fibers are not supporting for the longer duration, but for a small, shorter duration, but quick response, quick contractions, these are very, very useful. This is what the information that we need to understand. Okay. So generally the sprint runners, so generally they are running for 100 meters, 200 meters. Generally in those individuals, now these white fibers will be much more. But those individuals who are going for some marathon run and all, in those cases for longer duration they have to run. So generally their red fibers will be more developed. So it all depending upon our training and all, for what type of things we are getting trained. So accordingly this development also can happen. So in this way, so suppose you are an athlete, so you may better know about these things. Okay, next one. So with this, regarding that locomotion movement, and for the movement of individual or for the locomotions, these muscle cells are very, very important. These muscle cells in coordination with the skeleton, they are causing the movement of various body parts, and even they are causing the locomotion of individual. So till now we studied about how exactly these muscle cells are present, what are the different types, how they are undergoing contraction, all these things we have studied. And this muscle cell is connected with the bone, skeleton, bone. How is the muscle cell directly connected here? No, in between there are some connectors are there. What are these called as? So muscle and bone are connected through tendons. Tendon is also a connective tissue. Please understand this. Tendon is also a connective tissue. So muscle is connected to the bone through the tendons. So in this way, now contractions in the muscle, now what they will do? Now they are ultimately causing some changes in the bone movement. If the bone is getting moved, what happens? We can observe some movements in the body parts or changes in the body postures. Or if our legs are getting moved because of this, now what happens? So now it can also cause the locomotion. So in this way, skeletal system is also very, very important in the locomotion. And why it is called as a skeletal system? Because it is providing support to the entire body. That's why it is called as skeletal system. But please remember, before starting this, I am telling you very clearly, in the NCERT simply they have given the names and numbers, that's it. They have not given further information. So that's why I am also not going deep into this. Whatever is the NCERT content, up to that only I am covering here. Clear? And even for NEET examination also this much is enough. So now let us quickly go through this. So under the skeletal system we are talking about the bones and also cartilages. Bone and cartilage, both of them are again examples under connective tissue. Bone is a connective tissue, cartilage is also connective tissue. But the major difference between bone and cartilage is the type of materials that are present in the matrix. Between the cells, some matrix is present, ground substance is present. In the case of bone, it is having some calcium salts. Mainly it is having some calcium salts. Calcium carbonate, calcium phosphate. Likewise, some calcium related salts are present in this. So that's why the matrix is becoming very, very hard for the bone. But in the case of cartilage, it is having chondroitin salts. And because of these chondroitin salts, what happens is, though it is strong, still it can be pliable. So you can bend it. Cartilage, you can bend it. It will not break that much easily. Yes or no? Cartilage, you can bend it. So that's why it is a pliable. But bone is not pliable. You cannot bend it. If you are trying to bend it, it will break directly. So that's why bone is non-pliable. Pliable, pliable. Non-pliable is the bone, but cartilage is pliable. Okay, means able to bend. Cartilage is able to bend, but bone cannot be bent. Okay, this is some basic information that we need to understand. Next, we are going with the direct information. And anyhow, in this chapter, we are not focusing much on the cartilages. Okay, even though at some locations, if you're talking about the ribs and all, here is the sternum, at the back we are having the vertebrae and ribs are on the lateral sides, but the ribs are connected to the sternum through again cartilage. So under the skeletal support system, cartilage are also very important, but we are not focusing much on the cartilages. Our main focus in this chapter is on the bones only, so we are focusing on the bones only. Clear? And total how many bones are present in adult individual? Please understand this. Why we are saying like this? So newborn individual can have approximately 300 bones, but later on these things will be replaced with various other things, and later on some things are getting fused. So that's why we are not talking about the newborn individual or the child. Directly we are talking for the adult individual. In the human adult individual, we are observing total 206 bones in healthy condition. Clear? So now let us see. And these 206 bones, for our understanding and study purpose, we are dividing into two different types. 80 bones are present in the axial, main central axis. So here it is having the skull and then vertebral column, sternum and then ribs. And these are forming the axial main central skeleton. And it is having totally 80 bones. Along with this appendicular sidewise, what are these? So it is the pelvic girdle. It is the pelvic girdle that, and then these upper limbs, and then sorry, it is the pectoral girdle and the upper limbs, and this is the pelvic girdle and the lower limbs, and these will come under the appendicular skeleton, and it is having total 126 bones. Slowly one after the other we will go through. We are not spending much time, so observe carefully. Axial skeleton is having total 80 bones. In this, skull is having total 29 bones. Skull means hyoid bone and ear ossicles. These are also we are including under the skull. So total you are getting 29 bones. And then vertebral column is having total 26 bones. Sternum, only one sternum is there, and ribs are actually 12 pairs. 12 pairs means 12 into 2, 24 are there. Overall you are getting total 80 bones. Now first we are talking about the skull. Skull is having total 29 bones. In this 29 cranial bones, this is the cranium, right? So now this cranium is actually having 8 bones, and the facial bones, those which are making this face, it is having total 14 bones. And then hyoid bone, it is present in this neck region actually. There is one ear, and then ear ossicles, 3 on this ear and 3 on this ear are present. So total 6. This is the total count for the skull bones. This is the NCERT diagram. They have given different bones and names. So please try to remember this diagram with labeling. Clear? And now I will go one by one. Now first we will talk about the 8 cranial bones. How these are 8? One ethmoid bone is present, one sphenoid bone is present, one frontal bone is present. But 2 parietal bones are there, 2 temporal bones are present. But again occipital is only 1 bone. Add all these numbers, you will get total 8 cranial bones. If possible, try to remember these names, because in NCERT at least some of these names they have mentioned here. So please go through this. Next one we are talking here are 14 facial bones. What are these 14 facial bones? One mandible, lower jaw, and maxilla, upper. So 2 are there. So these are the maxilla, are the upper, and then these are 2 in number. Next one, inferior nasal concave, and the lacrimal bones, and then nasal bones, palatine bones, zygomatic, vomer. Even with the numbers also I have very clearly given. Only mandible and vomer are 1 in number, remaining all are 2 in number. So these are total 14 facial bones. Clear? Next one, so here we are observing one hyoid bone. It is actually present in this neck region. Clear? Next one is regarding the ear. So in the ear, in each ear, 3 here, 3 here are present. And what are these? And here these are also called as auditory ossicles or middle ear ossicles. Okay? And here these are called as malleus, incus and stapes. Okay, so these are the 3. So in this way, 3 on this side, 3 on this side, total 6 here, 1 here, 14 here, 8 here. So total you are getting 29 number. Clear? These are the skull, related to the skull. And now we are coming to the vertebral column. So just below the skull, here it is starting, and here it coming up to this tailbone. Clear? So total how many are there? 26 are present. The vertebrae that are present in the neck region are called as cervical vertebrae, and then thoracic region from here to here. So here total, please remember this number, 12 thoracic vertebrae, and how many ribs are present? 12 pairs are present. So one is connected to each one. So one vertebrae with on both the sides. So one pair of ribs with one vertebrae. So this number you please remember. So 12 thoracic vertebrae. So that's why in the next section we will study about 12 pairs of ribs. This number you need to remember. Okay. Next one, below this will be the lumbar region, and these are called as lumbar vertebrae, total 5 in number. And after this, one sacral, one coccygeal. Please remember this. These are in the adult individual we are talking, right? So actually here in the newborn individual, how many will be there? 5 will be there. 5 will be there, and these 5 will fuse and they will form one. So that's why sacrum is only one, it is fused. And coccygeal, in the newborn individual 4 are present, they will fuse to form only one. So only one coccygeal is present. So in this way, this number you need to add 7 plus 12 plus 5 plus 1 plus 1, you will get 26. Okay, don't count 5 and 4. Okay, because we are talking for the adult individual. This is just for information. In the NCERT they said sacral, one fused they give. Since they gave fused, how many are fusing here? 5 will fuse together to form this one. So in this way information you can understand, that's why I am telling you this. Clear? Next one, for these vertebrae at the center, for these vertebrae they are having some gap here, and this is called as a central canal. And what is passing through here? So anyhow, brain is present within this skull, cranium, right? And this brain is continuous with spinal cord. This is the central canal or the spinal canal in which spinal cord is passing through this. Spinal cord is passing through this. Clear? And for this spinal cord, again from the sides, again the nerves will come out, and these are called as spinal nerves. So regarding these things in the next chapter we will study. Cranial nerves and spinal nerves, these are the nerves we are studying. So spinal nerves are coming from the spinal cord, so they are coming out from this. So in this way, these vertebrae are indirectly, they are providing support to this spinal cord. Not only this, these vertebrae are also attached to this rib cage, okay, ribs. So in this way, so they have given some information. Next one, so between these vertebrae there is intervertebral disc. Actually this intervertebral disc is a fibrous cartilage, and actually it will provide some cushion nature also, so that it will take some pressure on it. So in this way all these vertebrae are protected from each other. Clear? Next one, anything else? That's it. So next one we are talking is regarding atlas and axis. Regarding these names, here they have not mentioned, but in the end of the chapter under the heading of joints they have mentioned about these names. What are these atlas and axis? For this cervical, the first cervical vertebrae is called as atlas, and it is bearing the entire weight of this particular skull and all. It is connected with that occipital bone through the occipital condyles. Okay, next one, second one is called as the axis. Between this atlas and axis we are observing a pivot joint, so we are able to move our head in different directions. Okay, anyhow these things we will study under the joints, but anyhow deep information was not given in NCERT, so simply remember this. So first cervical vertebrae is called as atlas, and second cervical vertebrae is called as axis. Simply these two names are enough, that's it, over. With this vertebral column is complete. Next one is the sternum. So this where these vertebrae are present on the dorsal side, whereas the sternum is present on the ventral side, but only one is present. And here what happens? Now ribs on the ventral side, they are attached to this sternum, but please remember they are not directly attached. In between this again there is a cartilage that is called as costal cartilage. Okay, so in the ribs portion it is present, that's why it is called as costal cartilage, but technically it is a hyaline cartilage. Okay, anyhow those names are not needed, simply remember this. Next one we are talking is regarding the ribs. So here these are the ribs. Please remember this is actually the hyaline cartilage. Hyaline cartilage present within the ribs is called as costal cartilage. That's why we are generally using a term called as costal cartilage. Total how many ribs are present? Total 12 pairs of ribs are present. One on each side are there, right? 12 on this side, 12 on this side. That's why total 12 pairs are present. That too I told you. How many thoracic vertebrae are there? 12 thoracic vertebrae are there. Now they will attach to this vertebrae. So please remember this. So in general we are talking about the vertebrae of the true ribs we are talking. True ribs we are talking. Vertebrae are present on the dorsal side. Sternum is present on the ventral side. On the lateral sides these ribs are present. And these ribs are actually flat like this. So on the dorsal side now they are going, and actually now they are divided with the two heads. Since it is on the dorsal side, now this rib is having two heads. How exactly they are attached to vertebrae, that was not given in NCERT. You don't need to worry about that, leave it. But simply remember that at the end it is divided into two parts. So it is having two heads, that's why it is called a bicephalic, means heads. So since it is having two heads, it is called bicephalic ribs. These are the bicephalic ribs. Please remember this. How exactly they are articulated or connected with vertebrae, that is not needed, so just leave that point. And now come back to this particular ribs. Out of these total 12 pairs of ribs, first 7 pairs of ribs, they are connected to the sternum through the costal cartilage. They are directly connected to the sternum through costal cartilage. So that's why first 7 pairs of ribs are called as true ribs. Please understand these names. It is the NCERT diagram. This is the NCERT diagram. So remember this. First 7 pairs. 7 pairs means 7 into 2, 14. So these 14 ribs. 14 ribs are out of 24. 14 ribs are true ribs because they are directly connected to the sternum. But what about the remaining 5 pairs? Please remember there is a problem here. In the NCERT they gave 8, 9, 10 as false ribs. 11, 12 as floating ribs. 11, 12 floating ribs correct. But if they are asking in examination, even in main examination also let them ask, no problem. But please understand, false ribs means not just these 3 pairs. You need to consider all the 5. 8, 9, 10, 11, 12, all the 5 pairs. 5 pairs means total 5 into 2, 10 ribs. These 10 ribs are false ribs. Please remember, many students are making mistake here. So please correct yourself. 8, 9, 10, 11, 12, all these 5 pairs are false ribs. Among all these false ribs, why they are called false ribs? Because these ribs are not directly connected to this sternum. They are not directly connected to the sternum. Then that's why they are called as false ribs. And even in this also, why they are rating 8, 9, 10 separately? Because these 8, 9, 10 are not directly connected to this sternum. They are actually connected to the cartilage of 7th rib. 8, 9, 10 are actually connected to cartilage of 7th rib. Understood or not? So in this way they are connected. So please remember this particular concept. Okay. So actually these true ribs, on the one side they are connected to vertebrum, other side they are connected to sternum. So that's why first 7 pairs are true ribs and they are called as vertebrosternal. One side vertebra, other side sternum, they are connected. So that's why they are called as vertebrosternal ribs, 7 pairs. But what about 8, 9, 10? 8, 9, 10 are not called vertebrosternal. They are not coming to sternum, but backside vertebra are there. Vertebro, and they are connected to this cartilage. That's why chondral. Cartilage, chondral. So that's why vertebrochondral. How many are vertebrochondral? 8, 9, 10 means 3 pairs, 3 into 2. 6 are vertebrochondral. But first 7 pairs are vertebrosternal. Please remember these names. Vertebrosternal are 7 pairs. 3 pairs are vertebrochondral. But what about the last 2? They are not even attached to this cartilage also. Simply they are present and they are stopped there. They are floating. That's why last 2 pairs means 4 in number. These are called as floating ribs. Names are very important. Don't get confused. So please understand 8, 9, 10, 11, 12. All the 5 pairs are called as false ribs. Among these false ribs, 8, 9, 10 are vertebrochondral ribs and 11 and 12 are floating ribs. Is it clear? So now this is the information. So please observe carefully. Now between these ribs again the muscles are present, right? So external intercostal muscles, internal intercostal muscles, many things are there. So in this way, ultimately what are there? So here on the dorsal side, vertebrae, ventral side, sternum, lateral side, ribs, all these are ultimately forming the rib cage. This is what. Clear? And now this thoracic cavity, the cavity enclosed by this is called thoracic cavity. And here on the ventral side, here on the posterior side, it is the posterior side, diaphragm is there. And all these are surrounded by the muscles. So in this way, they are making one completely closed system. Yes or no? So that's why thoracic cavity is a closed container. So that's why any change in the volume of this thoracic cavity is showing some effect on pulmonary changes, right? So already this information we studied in breathing and exchange of gases. Interlink this concept. With this particular part is completed. With that, axial skeleton is completed. Now we are going with appendicular skeleton. In the appendicular skeleton, total 126 bones are present. And in this, total 2 girdles are there. Upper one is called pectoral girdle, lower one is called as pelvic girdle. And then these are 2 limbs and these are 2 limbs. Total 4 limbs are present. So please observe carefully. 4 limbs are present. Each limb is having total 30 bones. So 30 into 4 total 120 plus pectoral girdle has total 4 bones. Okay, and the pelvic girdle is having only 2 bones. So in this total 126 bones are present. So first we will talk about the limbs. First we will talk about the hand, it is also called as a forelimb. In this total 30 bones are present. What are these 30 bones? One humerus is present, and then here one radius and one ulna is present, and then carpals. Carpals are also called as wrist bones. Actually they are total 8 in number. And then metacarpals, these are also called as a palm bones, total 5 in number. And the last one are called as a phalanges, total 14 are there. How? Only the thumb is having 2, but remaining 3 plus 3 plus 3 plus 3. So in this total 14 are present. Everything very clearly they have given. Phalanges, they are also called as digits. Okay, these are 14 in number. List, please remember as it is. Nothing more than this is required. Clear? And in the same way, one leg, one hand is having 30, other hand is also having 30. Names are same. Now we are talking for the hind limb, which is also called as leg. First we will see for one leg, same is the case for other leg also. First we are talking about this particular one. It is called a femur bone. And the femur bone is the longest bone. It is also called as thigh bone. And number is only 1. Next one, after this there is a patella. And this patella, actually originally it is not a bone, later on it is ossified to become a bone. But anyhow, under the name of the bone we need to count it. So it is 1 in number. Next one here, tibia and fibula, 1. Tibia 1 and fibula 1 is present in one leg. Next one are the tarsals. Tarsals are also called as ankle bones. Total how many? 7 in number. Okay, wrist bones are 8 in number, but ankle bones are 7 in number. That one number is covered with this extra one, that is called patella. Okay, and next one are metatarsals. So these are again 5 in number. Phalanges, once again these are the toes, again these are the digits, so they are again 14 in number. So in this way here also 30 are there. So each hand is having 30, each leg is having 30. So that's your total 120 bones are completed. Clear? And now we are talking about the girdles. Please remember, some students are making wrong calculation for the girdles. Okay, please remember this. Completely it is called as girdle. Completely it is called as girdle. Please remember this. One girdle. This is one girdle. Pectoral one girdle. Pelvic one girdle. Some two teachers and even students are also making wrong calculation. They are thinking off part as one girdle. No, off part is not called as girdle. Girdle means it is a complete one. That is called one girdle. So we have one pectoral girdle and one pelvic girdle. So in each pectoral girdle here, sorry, in one pectoral girdle, right? So in pectoral girdle again each half. Now here 2 bones are there. This is called as a clavicle or collarbone is there, and then there is a triangular shaped scapula is present. 2 are there. 2 on this side, 2 on this side. Total 4 are present. That's it. So regarding this collarbone, now it is connected to this particular scapula, and the scapula is on the dorsal side. It is a triangular shape, and on the upper this side it is having a process that is called as acromion process is present, and below this you are observing a particular cavity. This is called as glenoid cavity, and at this glenoid cavity what happens? This is the humerus, right? This humerus, head of the humerus is attached at this glenoid cavity, and here you are observing one ball and socket joint. Yes or no? This is the ball and socket joint you are observing. These are some information that they have given in NCERT. That's it. Very simple. You don't need to worry too much about this particular content. Simply observe what is the name, where it is present, how it is connected. That's it. Clear? With this, pectoral girdle is completed. So here on one side, here on one side. That's it. Completely it is the one girdle having total 4 bones. Out of the 4, 2 on this side, 2 on this side. Collar bones, and then this is the other one is the scapula. 2 scapula are there. That's it. Next one is the pelvic girdle. Please remember here, when we are talking about the pelvic girdle having 2 bones, it is in the adult we are talking. Because initially actually it is having 3 bones on each side. 3 bones on this side, 3 bones on this side. What are these 3 bones? Ilium. Spelling of ilium is very important. Spelling of ilium is very important. Observe carefully. I-L-I-U-M ilium. I-L-E-U-M ilium. Okay. What is this I-L-I-U-M ilium? It is the bone. It is the bone that is present in the pelvic girdle. But what is this ILEUM ilium? It is the part of small intestine. It is the part of small intestine. Small intestine has a duodenum, jejunum, ileum. This is the spelling of that ileum. This is the spelling of this ileum. These two are completely different. So please understand this. We are talking about the bone present in the pelvic girdle. Its spelling is ILIUM. Please observe carefully. So it is the ileum, and then this is the ischium, and this is the pubis. And initially 3 are present here, and actually these 3 will fuse and they will become one, and that one is called as coxal bone. This is one coxal bone, this is one coxal bone. So one coxal bone here, one coxal bone here, both of them combined they are forming one girdle, and this girdle is called as a pelvic girdle. Clear? And anyhow, it is on the back side. It is the sacrum and the coccyx. These are the ones that are connected here. Anyhow, we are not talking about this. This is the girdle. Clear? And here, this coaxial bone and this coaxial bone, and these two are connected here, and it is called as a pubic symphysis. And this pubic symphysis is once again, it is having the fibrocartilage. It is also again connected with the cartilage. Clear? So this is what about this particular pelvic girdle. In this pelvic girdle, here you can observe a cavity, and this cavity is called as acetabulum. And here this thigh bone is connected to this acetabulum, and here also you are observing ball and socket joint. Clear? This is how they are connected with the limbs. Clear? After this, regarding the joints, they have given some information, and actually joints, see, different bones are there. So as a whole, if they have to function together, they should have some joints, and at these joints again some connections will be there. Bone to bone connections, bone to bone, they are joined by, so they are joined by the ligaments, and bone and muscle are joined by the tendons. And now the movement of these will cause the movement of the bones, and because of the movement of the bones, now we can change our body postures, or even we can go for locomotion also. So these are some names that they have mentioned, some basic information they give. Simply I will read and I will complete this. Joints are the points of the contact between the bones. So two different bones can be joined, or between bones and the cartilages. Between the bone and cartilage also we can find these joints. Next one, force generated by the muscles is used to carry out the movements throughout the joints. At the joints, the bones, if they have to move, the muscle that is attached to the bone has to do some contractions and relaxations. Then only the movements can be shown here, where the joints act as the fulcrum. So around this particular joint, the movement of these particular bones or cartilages is taking place. The movability at these joints vary depending on different factors. So how exactly the arrangement is? See, all the joints are not working normally. So this is the ball and socket joint. So we can rotate like this, but this one, it is having only movement in this direction only. So in this way, there are various factors that will decide the movement of these particular joints. Next one, joints have been classified into three major structural forms like fibrous joints, cartilaginous joints, and synovial joints. In this way, based on the type of joints, we are talking like this. First one we are talking is regarding the fibrous. So between the two bones, there are some fibers. They are actually connecting these two. Generally, we are observing this in the cranium. Okay, in the skull, in the cranium, we are observing this. And generally, these are not helping in the movement of these joints. These are permanently attached joints. And these are technically called as sutures. These are technically called as sutures we are observing in this cranium. In simple, I am reading this. So you please go through NCERT each and every word. Next one is regarding the cartilaginous joints. So there are some locations where this cartilage is joining these things. If you are talking about the intervertebral disc between two vertebrae are bones. Between these two, now again you are observing this fibrous cartilage. Okay. So in this way, cartilage is again different types. Here we are observing fibrocartilage. You should not consider this under the fibrous joint. You should consider this under cartilaginous only. And next one, if you are talking, this is the sternum and these are the ribs. Again, these two, between these two also, cartilage is connecting this. That is called a costal cartilage in that location. So in this way, different locations we are observing this. Okay, but generally these anyhow fibrous joints, they are permanently fixed. They are not allowing any movements. But these cartilaginous joints, they will allow small movements. But these synovial joints, they are allowing the maximum movement at the joints. And here there is a synovial cavity will be present at these joints. Synovial cavity is there, and this cavity is filled with some fluid called synovial fluid, and different materials are present in this. They will help in the lubrication at these particular junctions. Many things are there. Like this, they are helping in the movement. Okay, articulation means the joinings. Okay. So this is what they are saying. So here again, within this synovial joints, again based on the type of these particular joints, again they have given the different types. Now let us quickly go through some examples. Ball and socket joint, that is present between the humerus and pectoral girdle. Here it is the ball and socket, one example they give. Hinge joint, knee joint, elbow joint, these are all hinge joints. Next one, pivot joint between the atlas and axis. Already I told, no, atlas is the first cervical vertebrae and then axis is the second cervical vertebrae. Between these two, pivot joint is there, so that it is helping in the movement of head in different directions. Next one, gliding joint between the carpals. So here wrist bones are the carpals, right? Here gliding joint. Next one, saddle joint between the carpal and metacarpal of the thumb. Here, So now this thumb is able to move like this, and this is the saddle joint. So in this way they have given some examples. So type of joint and example, at least NCERT examples are important. Clear? At least remember this. Next one, disorders of the muscular and skeletal system. Direct information, you need to remember this. Myasthenia gravis is an autoimmune disorder. Here it is affecting neuromuscular junction leading to fatigue, weakness, and then also paralysis of the skeletal muscle. Next one, muscular dystrophy, muscle dystrophy, damaged. So it is the progressive degeneration of the skeletal muscles, mostly because of some genetic disorders. Okay. So we studied about the myotonic dystrophy in the principles of inheritance and variation. Like, so in this way, there are so many genes that are causing these particular disorders. Next one, tetany is the rapid spasms. These are the wild contractions that are occurring in the muscle due to low calcium ions in the body fluid. Okay. Next one, arthritis is the inflammation of the joints. Try to understand the difference between arthritis and gout. Gout is also inflammation of the joint, but in the case of gout, the inflammation is due to accumulation of uric acid crystals. So in this way, so this arthritis is a general inflammation. Because of any reason this inflammation can occur, it is called as arthritis. And osteoporosis is actually age-related disorder. Mainly we are observing this in the case of females after the menopause, because after the menopause actually estrogen levels will decrease. So because of this decrease in the estrogen levels, now what happens is the bone density will decrease. Because of the decreased bone density, there are more chances that so it can undergo the breakdown or the fractures. So this particular condition is called as osteoporosis. Okay, osteo means the bone, porosis means porous nature, means weakness. So that it can break or it can undergo any fractures anytime. So this is what some hormone related information. So please remember this. So with this, the NCERT content is completed. And before closing this, once again I'm telling you, so these video lectures are available both on Live Prep app and also on YouTube channel. But the test series can be given only through the app. So with this particular revision part is completed. So in the next lecture, we can meet with a different chapter. Thank you.